On behalf of the Australian Plant Breeding Academy, it's my pleasure to welcome everyone to the second seminar in our series, titled Breeding for Impact, Navigating the Modern Breeding Pipeline. And so this seminar is a good opportunity for all of us to broaden our perspectives. And we also hear firsthand from a wheat breeder, Adam here, which is working in Australian Grain Technologies based at Roseworthy, South Australia. We'll gain insight into the transition from public research into commercial breeding, and to better understand the scale and complexity of modern breeding operations, and explore how commercial breeding is integrating advanced and cutting-edge technologies into their breeding programs, and what this means for the future of the industry. And just a brief background on our speaker today. So Adam is a wheat breeder, as I mentioned, with Australian Grain Technologies, and he completed his PhD with Julian Taylor, actually, which is there in the audience, at Adelaide Uni as well, where his research focused on improving genomic prediction accuracy and applying genomic selection in large-scale wheat breeding program. And since joining AGT in 2016, Adam has played a significant role in developing and implementing genomic selection approaches across their breeding programs. And his work sits right at the forefront of modern plant breeding. And he's deeply passionate about improving the efficiency of breeding systems to deliver high-performing wheat varieties that support Australian grain growers and rural communities. So without further ado, I will pass the microphone to our speaker today, Adam.
Thanks, Tim. Yeah, so my name's Adam. I'm from AGT. I'll just get this mic clipped up. So yeah, I've been with AGT for about 10 years and worked mostly on wheat breeding, but a bit also in bioinformatics and our genomic selection pipelines. So that's sort of been good for me, I think, because it's given me like a platform or a thing to talk about the broader AGT breeding programs with, rather than just my like wheat breeding program at Roseworthy. So I get to interact with a lot of the different crops, and we try and work as a pretty coherent team as well. So should just say, for the last couple of hours, I've been lecturing at the other end of the room. So two things from that: I might look a bit tired and just like fall over in a heap because I've just been talking for so long. And I might also be talking in a slightly condescending directorial manner like you do when you talk to students. Sorry, guys, but you're talking a different style of talking than a group like this. So I'll just take a few moments to readjust and change gears a little. Yeah, and definitely keep this conversational. If there's any questions during, just stick up your hand and yell at me. And the same online. I think you're going to check for any questions online. So if you're online, I was going to say, let us know if you can't hear us well, but that's pointless. Just ask a question anytime and we'll address it. So yeah, so what I'm talking about today, I'm not really sure. We'll see where the slides take us, but I've put together some slides, or put together / stolen, that sort of get it. I've got a bit on like the history of commercial plant breeding in Australia. And then I guess the theme is around how a commercial plant breeding program operates and how we do what we do. So that'll be the broad theme of the day. Oh, is it not working? Oh, there we go. Okay, we might go forward a few slides if that was a lag. So wheat breeding in Australia, if we go back in time 20 years to around the turn of the century, around 2000, this is what it looked like, right? So there was wheat breeding programs set up all over the country, and they were funded largely publicly. So they were housed either within state departments or universities, and they were funded through government, so federal and state funding, funded by the universities, and through like industry bodies like the GRDC. So in South Australia, you can see you had University of Adelaide, where we are now, and SARDI were based both here at Waite and at Roseworthy. So This is how things operated for like years and years and years before that, and it was all happy days. But there was a problem that was occurring, and it's not a very complex problem. It's pretty simple to explain. But I always think if you come up with an analogy, and it's a good analogy, which I think this one is, then take your time and explain something with an analogy. So I'm sure you'll all recognise what that thing there is. Is it? Yeah, hands up if you know what it is. Keep your hand up if you knew what it was or would have recognised it more than a month ago. Yeah, my hand's definitely down there. So what used to happen in the world was there was oil being produced from these wealthy nations in the Persian Gulf, and the oil would flow happy, happy, happy through the Strait of Hormuz onto the rest of the world, giving us all that oil goodness that we need. But there was a problem. The oil stopped flowing, right? Some crazy orange man did some things, tweeted some stuff, and the oil stopped flowing. So where's he going with this? How does this relate to plant breeding? So what used to happen under this model with like breeding programs being funded by largely the government was there was funding. Forget about the Gulf of the Strait of Hormuz now. This is just funding. And it used to flow, not through the strait, but it used to flow out to the breeding programs. And that was great. They were all getting funded. But the funding stopped. So why did it stop? Well, basically, Australia is a country with not that many people, and we produce a lot of food. So there's no food security issues here. We've also got a population which is like largely in the cities. So you don't get many votes for funding like tomorrow's food production or research that drives that. For that to happen, you basically need food shortages, and that's just not going to happen in Australia. So it's really interesting looking back at this. I was actually, around the time I was starting at APBA, I was in the library here at Waite, and I found like the box of books that they're giving away because no one wants to read them. And there was a conference proceedings from a wheat breeders assembly in the 80s. And me being the nerd I was, I picked it up and had a look and took it home. And yeah, in one of the topics for discussion at that wheat breeders assembly was, what are we going to do about this problem we've got with the lack of funding? So this is back in the 80s. This is 20 years before this started. And like me at the time, I was amazed that this was bubbling away in the background years before anything actually happened. But what ended up happening was the people in the industry, or the industry at the time, set up a system for endpoint royalties. So there was legislation around plant breeders' rights that was put into place, and then endpoint royalty, mechanism was set up. So you might have the legislative protection around your variety, but you need the mechanisms in place to actually collect that, so that plant breeding could fund itself, basically. So farmers would, when a farmer grew like a ton of grain that was bred by someone, they'd pay a couple of dollars back to the breeder of that variety, and that money, that endpoint royalty, would fund plant breeding. And that's the system that we have now today. So that's worked really well in Australia, largely because we're lucky in the respect for crops like this, like wheat, that are it's largely export driven. So almost all of the crop is exported, or 3/4 of it, and it's exported by not that many parties. So you can talk to those parties and get them to collect the APR, and you've captured like 80% of the crop. If you go to Europe, that's very much not the case. You've got to talk to like hundreds and hundreds to get to half of the crop. So it's a little bit more complicated. But back to what happened in Australia. So these orange boxes on the left, they're the old pre-existing breeding programs. They were privatised. So this happened through the endpoint royalties that were there in place to fund them. And then the GRDC helped push things along and put money in to set up private companies to use that germplasm to do plant breeding. So you can see, especially in the early years, it was a bit of a mess. Like there was a lot of action happening with people getting involved and going out, and germplasm moving from program to program. But you think about that from the breeding point of view, this is a bit of a sidetrack, but it's like what I was talking about this morning with genetic variants and crossing across programs. There's all of this mixing of genetics that was housed like within these programs, and there was a bit of that before, but probably less than what happened after. And now you look at within AGT, within one company, you've got germplasm from a few different breeding programs that's come together in that one company. So on the right, you've got the major plant breeding companies in wheat breeding in Australia. And then some newer entries down here. So RAGT have recently bought BASF, which came out of the CSIRO program. So they're active in breeding. And then you've got some smaller players over here. So there's companies ranging from Rebel Seeds, which is like basically a grower cooperative, releasing the variety straight out of Simmet, largely. And then next to that, you've got GDM, which is like the other end of the scale. They're a massive plant breeding company in South America, largely in soybean, I think. They're like dominant in soybean, and they're getting involved in wheat breeding in Australia now. So the point of that is, what's happening on the right-hand side there now is exactly what I think the industry wanted to happen in setting up EPRs, in that you've got a highly competitive landscape that all of these companies, like none of those companies are going broke. They're all doing well. They're investing more money into plant breeding, and that's great for Australian farmers. So that's a positive story, I think. Sounds a bit like propaganda. So zooming on an AGT, so I've got a few slides on just who AGT are, but I thought I'd just start with our company motive, or what's our goal, our purpose. So I've got it written down. We've got it written a lot of different ways because how it's written doesn't really matter. It's more the content of the words that matters, or the message. So crop improvement, for stronger rural communities, and improved global food security. So that's really important to us. Like that's the same purpose as the purposes of those original plant breeding programs that were publicly funded. And I think that's really important, is that although the structure of how plant breeding is carried out in Australia is different to how it was, the purpose is still the same. So, and I guess really importantly for us as well as a company, is that purpose isn't to generate profits for shareholders. We do generate profits for shareholders, but the main purpose of those profits is to feed back into plant breeding, to keep funding what we do, and to keep growing and coming up with better ways to do stuff, and keep reinvesting those EPRs into plant breeding. So that's really important for us. So AGT is a national company. So we were formed out of that mess that I had a few slides ago. Largely started in wheat breeding, and now we've got crop breeding centres spread around the country. We've recently just tried to switch internally from calling these nodes to crop breeding centres, and it's a bit of a tweak in the old brain. We've been calling them nodes for 15 years. So the crop breeding centres, we've got Narrabri up in the north, so that's up here. So you can see there we've got around 10 people. And the crops that we breed out of there, or the crops that are led out of that program, are spring wheat breeding for the northern region and durum wheat breeding. And then we also do some barley testing. We've got a durum quality lab set up there, so that's some additional testing that happens at that node. And we've got variety support people. So variety support's like the marketing people on the ground that go to field days and manage seed production. The Eastern Crop Breeding Centre at Wagga Wagga. We've got spring wheat breeding and winter wheat breeding. Got additional barley testing and canola testing, and also variety support people there. At Roseworthy in the south, the breeding activities that happen there are spring wheat breeding. That's my program, along with James Edwards. And then we've got barley breeding program. So this is a nationally focused program that's based at Roseworthy, and the same for canola. And then we've got durum wheat testing. So durum's grown in South Australia, but the breeding program's led out of Narrabri, and the variety support. And then at the Western Crop Breeding Centre at Northern, we've got spring wheat breeding and lupin breeding. And then we've got barley testing and canola testing that happens to support those programs that are based at Roseworthy, plus the variety support activities that happen there. At all of those nodes, there's like quite a large amount of field activities. So that's probably half of what those 10 people are, or probably more at each node. And you can see at Northern, Wagga and Narrabri, there's around 10 people. And then at Roseworthy, there's 60 people. So that's obviously quite a few more. Part of that is because there's these centralised breeding programs in barley and canola that are just based at Roseworthy, and that brings additional people. But we've also got a lot of centralized activities at Roseworthy. So Roseworthy is like the hub for a lot of activities. And some of those are wheat quality lab, wheat, barley, and canola. So basically everything except durum testing is centralized at Roseworthy. Glasshouse activities. So this was a big one we changed around 10 years ago or 12 years ago. All of the other crop breeding centres used to have their own glasshouse or glasshouse facilities and used to do their own crossing. And we thought there's two challenges with that, right? You've got a really skilled work base. Anyone who's done crossing in the past, even for a crop that's relatively easy, it's quite a skilled task. It's pretty specialised. And a crossing block happens twice a year. So you take people from anywhere, normally from the field team or whatever, go into the glass house, do crossing for a month, and then come out and drive a tractor. And that was great while we were small, because you want people who are like cross-trained really well. But then we got bigger and we needed, I guess, more people and more dedicated people to try and innovate and do it better. And we also needed better infrastructure. So we wanted to invest in like a state-of-the-art glasshouse. And it doesn't make sense to have 4 state-of-the-art glasshouses spread around Australia that are all partially used at different times of the year. You're better off just to have one massive one at Roseworthy. And that's what we did there. So we centralised that at Roseworthy a few years ago. Connected to the lecture I was giving this morning around genomic selection. So that's really led to a need for different skill sets in the plant breeding company. So this bioinformatics line, I guess 10 years ago when I started, that was like a third of my time. Before that, it was Hayden, part of his time doing it. And now we've got 4 people full-time in that space. So that's quite a change, led largely by genomic selection and more marker data that we're dealing with. So, you know, the marker data gets used in genomic selection, but it gets used everywhere, basically. We use it in all of our analyses that we do. We use it to correct pedigrees and check things are right. Manage seed inventory when you get to commercializing a line. There's huge amounts of risk if you release the wrong line. So we use that to trace identity of our lines throughout the breeding program. So the business model of this is not AGT specific, this is any commercial plant breeding company. You've got value creation and value sharing. So you're probably all familiar with the value creation side. That's your typical breeder's equation. And it's directed by breeding objectives. So you need to, like any breeding program, you need to clearly set your breeding objectives. And then you're trying to increase the amount of diversity in the breeding program. The accuracy, you want to obviously be really accurate in what you do and do it at large scale. And you're trying to decrease the generation interval over time. So that's how we create value. That's all within one crop. We want to have bigger impact, do it on more crops. And then if we want to actually make use of that value, you could have like the best varieties in a given crop. But if you don't produce seed that's clean or tell anyone about it or tell people why they should be growing it, and no one grows them, well, then you haven't actually done anything. You've just got this good variety that's sitting on the shelf. So this is actually a really important part of the business still, is that we need to work on the variety adoption rate by, not by like snake oil salesmen forcing people into or tricking people into growing them, but by just effectively communicating the reasons why they should. And to do that, you've got to have, you've got to be really honest and have a good amount of integrity. And if your variety isn't one someone should be growing, you tell them that. Because that means when you go and tell them the variety they should be growing and it is yours, they can believe you because you told them last time not to grow theirs. So all of that produces value that's realized by growers. Some of that value comes back to us in the endpoint royalties, and that's value sharing, and that goes back into value creation, and the cycle continues. So happy days. This breeding objectives and genetic gain, or the breeder's equation model, that's what we're going to talk about a bit now. So value creation, let's break down, I guess, the parts of this equation and talk about what they are. So the breeding objectives, what we're really trying to do there is ensure the direction of the breeding program aligns with what growers want. And growers, it can be finicky to nail down what they want if you get into specifics, because they're reactionary, right? Based on what they saw last year often. But at the core of what they want is increased profitability. So how we define profitability, I guess, is grain yield times by the price you can get minus the costs. And then around that, there's some risk factor. So we're trying to increase the amount of grain yield they can get. That's obviously, that's a pretty clear one. Maximize the price they can get for selling that grain. So in Australia, in wheat, there's the classification system where you want to, growers can achieve a higher price by delivering prime hard or Australian hard varieties. But there's risk there that they might not receive that grade if, for example, the variety had high screenings or low test weight or was low protein. So you want to minimize those risks. And then there's management costs. So the big one there is, disease resistance, herbicide tolerance, sort of comes into that. So that's sort of across the board, I guess, where it can also impact with grain yield by controlling the herbicides better. So all of these things we're trying to work on at different levels. So scale, larger population size increases the chance of finding desired genetic combinations. So a couple of key words in that sentence are desired genetic combinations. So there's multiple genes we're targeting here. It's not just one or two, and chance. So we're not like the GM and gene editing is still in a research phase. There's no like commercial varieties out there with that in there, and we're not using it on a day-to-day basis. So that means the type of genetic variation that we're working with is not directed. With gene editing and GM, you can direct the type of genetic variation that you create, which means you're not just relying on chance, but we are just relying on chance, which means the scale of what we do is really important to just increase the chances of finding those good genetic combinations. Accuracy, so signal versus noise. You guys could explain this 100% because you're in my lecture earlier. Happy days. So yeah, this is looking at, when we see plots in a paddock, we want to make sure we're selecting a high yielding plot because it had good genetics, not just because it landed in a good part of a paddock. So that's more around technology. Diversity is an interesting one. So we need to balance diversity with genetic gain. So breeding programs can suffer either from spending too much of their resources on bringing in diverse material, not getting rid of it quick enough. And they can also suffer by not focusing on diversity at all and losing diversity. Then you've got no diversity to select within to make genetic gain. So we need to balance those two things. And it's important to keep in mind, like, we need diversity, but most diversity that you bring in will be bad and you'll be trying to undo most of it. So we need to balance that. And then time. So this is the generation interval. And that's like turning the wheel. So that's how quickly the breeding program's turning. And there's a trade-off there with accuracy. So it's really easy to just increase or decrease the generation interval and go quicker. It's hard to do it while maintaining accuracy in what you're selecting and what you're using as a parent. So the breeding objectives cycle, I guess, is we set the breeding objectives and then we push those breeding objectives into the program. And the breeding program does its thing, executes those objectives and creates a product. So that's the product. And then we release that to growers. Those growers, grow the variety, form an opinion on it, find what's good and what's bad about it, find what matters to them and what doesn't matter to them. So there might be something that's really good in that variety, but they just don't care. It's not really a positive, and vice versa. So then that feeds back to us the information around what growers want. And then we filter that through what's possible, what we think we can do, and that goes back into our breeding objectives. So this cycle is a continuous thing. And we put a lot of effort internally on this part. So we spend a lot of time actively seeking information on what growers want. We've done surveys before. Probably the best way is just having a strong network of talking to growers and agronomists and filtering what they say. It's easy to talk to one or two noisy growers or agronomists after something that's happened, like a rust epidemic in 2022, and think, oh shit, I need to chuck away like half my breeding program because it's not good enough for rust. And then 2 years later, they haven't seen rust for 2 years and they don't care anymore. So the hard part with plant breeding is you're breeding for what farmers want in 10 years' time. So you can't be too reactionary. You want to just steer it in the right direction, but not make sharp turns. Otherwise, you're just left with this this gobbledygook program. So breeding objectives. What I talked about then was us gathering information from growers and filtering down it down into what our breeding objectives should be. So we also go through this process of sitting down as a science and VS team. So all of the breeders, all of the science-based people in the company, along with the marketing and variety support teams, sit down and go through each program, each trait that might matter, and think about what a target performance could be, and then what the proportion of our germplasm that we want to see with that target. So it can't be 100 for everything, because if it's 100% target for a high performance level for all the traits, then the sacrificial lamb will be grain yield, because that's the last thing, right? So you need to make compromises. But we put a lot of effort into doing this, and you can see it's highly top secret. There's numbers behind this, but I stole this from a talk Hayden gave, so I thought I've got to keep that special sauce within ADT and keep a bit of intrigue around what we do. But we go through those numbers quite often and spend a lot of time on it, and the breeders really look at it and pay attention to how they're tracking against what we said was important. So a breeding program is a constant place of tension due to limited resources and seed quantity. So that tension word is a really important one, I think. It's like that tension I was about before around the breeding objectives. There's tension between them. You can't have it all. In a breeding program, you can only run so many plots. So sometimes the answer is to run more plots, but it costs more money. So you can't always do that. You can't always run more plots because you might be limited with seed and you need to test other things. So you're trying to find this perfect compromise or the best compromise. So some of the solutions that we work with are just bulk scale, invest more each year. If you're running the company, that's an option you have. You could try and just brute force it, not do anything better or smarter, just hire more people and run bigger programs. And that's a real strategy. Like there's strong merits for that. I've got there as well more quarantine space, so bringing in more diverse material to have like better or more capacity to evaluate that. Or you can invest in more capital to try and increase efficiency of the team. Some of that capital might be a result of a project where you've come up with a new way of doing something or like innovated. So you can have better equipment with increased throughput and accuracy. Or there's innovation and improvement. You see there's a lot more dot points here. And a lot of what we do, I think, like there's within ADT and the breeding programs, obviously part of what they think about on a day-to-day basis is turning the wheel, just running the program, do your selection, make your crosses, focus on your traits that you've said are important, just keep things ticking over. But another large part of what we're doing is thinking about how we can do things better. Like, how are we cleaning that seed? Is that quick enough? How do we, like a big one 5 or 10 years ago was how we count grains was quite slow, or measure screenings was quite slow and quite noisy. And we started using robots, so we wanted to be able to load variable sowing rates within the robot. So we had to come up with a way to count grains and measure the 1,000 grain weight of our varieties. So we came up with a way that worked alongside our cleaning process and basically adds on 0 time to that process of cleaning the grain. So that was a really important innovation. And there's heaps of things like that breeders and the breeding teams are thinking about every day. Like it's not just once a year we have a bit of a think and try and come up with some new ways to do stuff. It's really core in what we do. So I thought I'll also put this up. So this is another example of tension. So we've got pros and cons of running a breeding program that's highly flexible, that's highly rigid. So if you think about a flexible program, it's one that, you've got stage 1, stage 2, stage 3, stage 4, and every year there's different numbers in each stages. Where the stage 1 lines get yield tested is different every year. Everything's always changing. So that's highly adaptable. That can be a good thing. But the bad side of it is it's generally less organized. Like there's like fewer systems to it. So it's less organized, it's harder to manage and it's harder to learn. Like if you've got a highly organized program and someone comes in and tries to operate it or work in the field team alongside it, uh, there's a pattern to it. So it's recognizable. They can pick things up and expand on it. But once they learn it, there's very little communication required because it's just obvious how it works. Whereas if something's different every year, there's just all these inefficiencies around always having to communicate what you're doing, where's this trial going this year? It's more error prone because there's nothing's normal. So it's quite a challenge. And then you look at the rigid programs. So the other side of that, so it's the same thing, just in reverse. They're generally more scalable. If something's really organized and structured, it's easier to scale that up and make it bigger. They're more intentionally designed. So in a highly flexible breeding program, you might, if you're trying to manage that breeding program, say, and you see the breeders made changes to it, some of those changes might be really sensible, well thought out, like good changes to make. That was a problem with the old program and they'd fix something. But then next to that, there's all these other changes that are just random. And it's like trying to pick up a genetic signal of a high yielding variety in a plot that's, got good and bad soil type. That signal to noise ratio is off. So it's hard to recognize what's an intentional decision and work out why someone's doing something. So a rigid program, when you make a change, you make it for the right reason and you stick to it and you give it the chance and you only change if that information changes. The downside of it is it's less innovative. You can very easily in a rigid program get caught in this trap of, why do we do something? Why do we do something this way? Well, it's just because it's how it's always been done. And I've never thought about it. It's been done like that since I came on board and I don't know, let's just do it again. So that's a risk. So you can see there's more dot points in this one and this one. So I think I've communicated there that AGT definitely sits more on the rigid scale. We try and keep things pretty organized, but we've got to be really conscious of these parts and try and keep the best of both worlds happening. And that's where having that structured review process for the breeding objectives is really important. We also, once a year in our meetings, just go through someone's breeding program and pull it to bits and critique it and think about could you be doing this? Why are you doing that? And really look for opportunities to improve. So we're definitely more along this side, but we've got to be conscious of the trade-offs that come with that. So I thought I've got an example here just of managing big data. So this comes back to that bioinformatics team and the new skill sets that are required in a plant breeding company. So the process for capturing genotype data within AGT is to book the job in, so provide tracking information to the lab, how many lines, date, what you want running it. That goes to the lab, receive a raw data file back, and then you clean and process that data file into a standardized numeric format. So there's obviously multiple batches of data coming in. So you receive the data back in AA, TT, etc. You need to convert that to a numeric format, but you need to do that in a systematic way so that next time you get a data file back, you do it the same way. And you don't have a -1 that actually is actually a 1. So you need to have a very structured process for that. Then we check pedigrees and identity and impute missing data and add it to a master database. And we've also maintained an ID database where we keep every instance of a variety that's been genotyped to check the ID is correct. And we can run things through that database like affiliate seed production lots to make sure they're selling the right variety. So that process is run through around 100 times a year in AGT, the process of booking in jobs and receiving chip data. And the database now across all of the species is around 1.4 million genotypes. There's almost 1 million genotypes in the wheat database. So we've recently had to reformat it in terms of what software we're using to store all that because it was getting too big to capture. But that's across 5 species. And if you multiply that out by the markers in each crop, there's 21 billion data points. So that's a heck of a lot of data to just maintain on storage devices and, you want to keep the structure there and keep all of the data accessible, but you still want to be able to just quickly jump in, grab out 5 lines and check something. You want it to be usable. And then what we do with that data, we've got GS calibrations. So we maintain around 200 prediction calibrations per year. So that's like trait by crop cases. Marker-based analyses, I don't have a line for that because I didn't get around to working out how many analyses we do with markers each year. But I imagine it would be around the same number. There's probably more actually, because there's a bit less trait, like a few less traits, but not that many more. But there's multiple trait analyses per trait, depending on the timing of when data is coming in and what selection cohort. So that stuff there, like that's what those 4 bioinformatics people do. And then we've got genomic predictions and managing selection indices. So we predict around 100 cohorts or batches of predictions per year. So you can see even just turning the wheel in this space becomes quite a job. And then in all of this stuff, there's trying to keep up with the size of a data set, and you call it innovation, but it's really just keeping up, where we've had to reformat the database. But there's also like new literature on better statistical models for traits. There's all sorts of research going on that we can either, at the very minimum, we want to be passive users of that research, where we, assess what's happening in the literature and use what we think is good. And what we actually want to do is participate in that research and help drive it forward, because you get a lot more out of it that way. So there's a lot happening there. And that's just I just thought I'd put that up as an example of one of the areas of AGT where there's a lot more scale happening now than what there used to be. How are we doing for time? For a few minutes? Yep. So this is just a similar example, but a bit broader and across the timeline of AGT. What sort of innovations or technologies have we implemented or developed in-house or brought on board? So let's call it our tech stack. So in the early days, things like summer nurseries and marker-assisted selection, logistics-wise, so we've got barcodes and databases were used. And you can see on the breeders equation, we've got a color scale. And then some of these are all one color, and some of them are a mix of colors. So summer nurseries is green, that's impacting the time, because you're just pumping through generations over summer to get to fixed lines. So you can see where we're having an impact with these things. Proprietary software handling systems. So the flow of genotypes or samples from one season to the next is really important. So we've got barcodes, but you need to be able to scan them, to weigh them if you don't have onboard weighers. And then that identity, some of them will be culled. So you need to be able to scan it and tell you whether it's keep or a cull. And if it's a keep, what trial is it going to and what's its entry number? And then print out a label to stick on a bag. So all of the lines we get obviously came from the previous year of trialing. So that's a huge amount of work at this time of year or in the last few months to manage that flow of samples. GPS guidance on tractors means more accurate plot work. Pedigree analyses and sparse designs. This is what I was talking about this morning with relationship-based analyses. Sparse designs is just spreading the replication that you've got over more environments to get a prediction of more environments, pick up more G by E. We transitioned to Limograde Marker Lab to increase the scale of what we could do there. So that's a grey one for scale. Robotic seed packing means better designs and more accurate or like variable sowing rate. So you can, or variable sowing rate by volume, same sowing rate by seed density. So more accuracy. GPS trip seeding means sowing more plots per hour because you can drive faster and spend less time turning around. Overdrive or rapid back crossing, brought that on board. Onboard weighing of harvesters, image-based grain assessment, more equipment for more yield plots. So that's just brute force, increasing scale. Genomic selection impacts sort of everything. Limograne club agreements. This is like a germplasm sharing agreement between AGT and all of the Limograne breeding programs around the world. So that's like we can access germplasm for crossing. And we've gotten a lot out of that in terms of, for example, septoria resistance, which is a bigger problem in Australia now. But it's been a big problem in Europe for a long time, and they've got high levels of resistance bases. So we brought in a lot of material and put a lot of effort into making crosses to that material. Glasshouse and controlled environment facilities, and then dedicated bioinformatics team. So this was around the time that genomic selection work had started, and then it was scaling up in terms of the amount of work that was being done. RGA, so that's like speed breeding or rapid generation advancement. In-house machine learning for work like what Tim's doing. And then there's a few other things that I thought I'll just call them top secret. So there are actually things there, but the beauty of this slide is they might not be things. They might just be just things I put there, and you're like, oh God, they must be working on exciting stuff. Okay, I think we're almost there now. So this is the breeding program. I was explaining this morning as well. So you're probably all familiar with it, but the general structure of a breeding program is to start with a lot of genetic material. So you've got large population size, but the unit of testing, or the amount of testing per unit per line is not very wide because you've got so many lines. So this triangle over here represents how wide the testing is per line. So when you've got a lot of lines, you're not doing much testing, and then you cull down to like advanced varieties, and you're doing a lot of testing. You want to pick off all of the traits that matter, and you want to like extend into more environments and make sure you're not missing anything. So if we think about, with all of the stuff that we've talked about over the last 40 minutes, this structure of the program, the scale is not quite right. So now more what we're doing is increasing the population size in the early generations like more relative to the size of the rest of the program. And that's largely due to GS, where, like I was saying this morning as well, you've got genomic selection, calibration, or predictions right at the start of the program because you don't need much seed to test it. And when you get that, you get a prediction for all of the traits. So you can build that into a selection index and do some really like powerful selection and cull harder. So this size of the program is still the same. We're just starting bigger and culling down to that. So it's larger scale and larger selection intensity. That's that. And then what that leads to is also if your selection is more accurate, then the whole program just becomes more efficient. So there's less things getting through to stage 3 and 4 that were like misidentified. Because if you're not using GS, so you don't have a prediction for all of the traits, you're doing tandem selection. Back in the day, we were stuck with worse analyses. You have escapes essentially, and you get to stage 3 or 4 and realize something's like not that good when you thought it was really good. Also, just the improved efficiency from having genotype data present in the breeding program is the first time we genotyped the stage 3s and 4s, the advanced material, we're finding these lines that had like high correlations. We're like, oh, what's going on there? These are like sisters, but they're 0.99 correlated with each other. So they're actually genetic duplicates. They were like derived from the same plant. And I remember it was one from 2 years earlier, and I was showing Hayden, and he said, yeah, I remember those 2 sister lines. They're in stage 3, and we were really like pulling our hair out trying to separate them and work out which one to take forward. It was like no wonder because they were the same thing. So that's just an example where those things sort of sound a bit innocent and innocuous, but that's an entry in the stage 3 that didn't need to be there. Like you're testing the same line twice. And those things just add up over time. Like the cost per line of a stage, like this is the stage in the program where you've got 100 genotypes. So you've come from like 20,000 plus down to 100. The cost of having a line in there is huge, not just on the amount of testing, like how wide the triangle is at this point, but the opportunity cost, because there could be something else up there that should have been in there that wasn't. And probably what it was you go through your selections, and if you're trying to get to 100, you go through and get to 140, and then you try and like really get it down to the 100. And you have these diversity crosses on the side that are like an introduction, and you try and keep them there because you're trying to feel good and like keep the diversity alive. But when it gets to that last cull, like they're the things that die. So it was probably an intro that could have like contributed to that diversity that we need in the programs, that it wasn't there anymore. So then with all of that information, we can fast track releases. So we still keep the structure of the breeding program the same and quite rigid, but we have like dedicated, like different paths to fast track varieties out to release. So while the standard structure of the program is 10 to 12 years to release, if we want to go really fast for a dedicated purpose, we can do it in like as low as 6 years. And we did that with a barley release recently. And then what's really exciting at the moment is getting into the space of like GS crossing, where we're trying to use things straight out of crossing, run a chip and predict them, and put them back into crossing as parents. So turning that wheel really, really fast. And obviously, there's a lot of things to work through there in terms of prediction accuracy, maintenance of prediction accuracy over time, but also how, like, just do they come out into the rows, or do you bulk them in a population, or how do you manage those things coming out? So there's a bit of a redesign of the breeding programs to work through that. So that's all. There's a photo of our glasshouse at Roseworthy. There's actually, at the moment, there's a shed being built here and another glasshouse out here. I think I've got that orientation correct. So that's expanding, and that's one of our wheat and barley yield sites from Roseworthy a few years ago. So there you go. Thanks for listening. Questions?
Thank you, Adam, for providing that valuable insight into the breeding program at AGT. Does anyone have any questions for Adam? I mean, I'll start from down the back because that arm went up first. Oh, sorry.
That was very nice, thank you. So in a hypothetical world, or perhaps a future world where GM and gene editing are completely legal and available to you to use in your breeding programs, how would you use them? And would you use them more or less than your traditional sort of scale random approach?
Yeah, good question. So that's one, I think at the moment, if you look at like, you know, a good company will have like risk mitigation strategies, or they'll be aware of what the risks to their business are. And one of the biggest risks for any plant breeding country in Australia at the moment, or in the world, is a technology comes along like gene editing, and it can lock away traits and push the existing companies out of the market. So everyone feels like they should be using GM or gene editing. And I think at the moment we're all struggling to come up with use cases or traits. Like we understand the concept around how you would use it, but to do gene editing, you need not just the trait that's a target, but you need the gene, and you need that sequence to be able to like hone in and target it. So if we can do that, if we can find those targets, which we've got a few of, we've got quite a list, but it's harder than you would think to come up with that list of targets. Then it comes into the breeding program, and this is probably where, like, I don't have it really clear in my mind. Do you generate the event once and then put that into the breeding program and let conventional breeding do its thing? Or do you do it the other way, where you breed these varieties and then like gene edit in at the end and just like boost your variety up? And that'll depend on like, you know, how hard it is to do that, what the like success rate is, how quickly you can bulk up seed again, where it needs to happen, all of those questions. So there's a heap of stuff we don't know. But I think the biggest thing we can do, like the gold standard, would be find variation for traits that we don't yet have. So create meaningful novel variation. So something like frost tolerance, for example. If you could find a way to find that target and create that variation, then like that'd be a game changer. Yeah.
Yep.
Thanks for your talk. That was very interesting. Just out of curiosity, when you get to a new variety trial, how many growers would try them, in how many environments, and for how many seasons?
Yeah, good question. So I didn't explain that very well in the talk, but you're referring when you get to MBT at the bottom, is that right? So the process, if I go back, so stage 4, that's internal AGT testing. That's around 50 environments across Australia for wheat, different numbers for different crops. NBT is a like industry funded independent variety trial. So that's not with growers, it's still plot trials, but it's just not run by AGT. It's run by GRDC as a breeding programs contribute to them. All up, there's probably like a couple hundred wheat sites across Australia, but you nominate into regions. So you might say this variety is a fit for Western and Southern Australia. I'll nominate into those. After that, like we'll send seed out to grower groups to do, they might do strip demos or their own sort of plot trials. Growers generally buy like a small amount, plant it because they need a bulk up seed anyway. So they'll normally plant it next to their existing variety. We'll tell them that's terrible because Jules has left the room now, but for statistical reasons, that's not good, right? Just plant one variety and then one variety. But they'll always do it. And then they'll bulk it up from there. Yeah. So it's a few year process to go through that.
Yep.
Thanks for the talk, Adam. I wanted to ask if you're able to share how long it took for AGT to be confident in adopting genomic selection, that journey, and then along that journey, were there any particular sort of outcomes or findings or results that gave you that confidence to adopt that technology?
Yeah, good question. Yeah, I think you asked it as for AGT to be confident, which is interesting. You think like for Hayden to be confident, and for me, the student working on it to be confident was pretty quick. I mean, I had like Hayden's guidance. He was probably influencing me a fair bit. Some of the more conservative breeders, I'll let you know in 5 years' time, they might still not be confident. No, it was relatively quick. And part of the main learning was it's really hard to assess the accuracy because you scare people off by saying, I've done this validation study where I've predicted these lines in a blind set, grown those lines in the field, and it correlated 50%. Okay, 50%, that's not that good. What are you talking about? But the problem is you're assuming that trial that you ran is gold standard, whereas that might have been, say if it even is 2 years, like there's error in that trial. it's not a full representation of environments. So the challenge is like picking apart what's actually prediction accuracy as where like you've actually mispredicted the genotype versus what's just a difference in environments. So in this prediction model, we've got say 10 years of phenotype data and you're predicting out and correlating against something you've grown in one environment. So it's not going to be a perfect correlation, but you've got to look at like how representative that is, and that massive genotype data or like phenotype data behind it is a more stable, robust, representative measure or set of yield data, I think. And then particularly looking at early generation stuff, like the first place for yield we were trying to use it was early generation, where at the time we were running, we called them half plots. So you've got enough seed to grow one plot. We didn't randomize it because we wanted to see families next to each other. So it was intentional to not randomize it. And the heritability of those trials was like 0.3, and it's at one environment. So you've got a set of environments, which is one, and a 0.3 heritability on that. So it's pretty easy to beat that. Plus, like I spent a lot of time this morning talking about multiple traits. So we can also get caught thinking, to use GS for yield, I've got to beat yield assessment, like just yield assessment. But you also get like maturity, you get disease, you get grain size, test weight, and you can build all of that into a selection index. And like I was explaining this morning, it's been literature around, selection methods and the fact that selection indices are a more efficient way of selection than tandem selection. Like that literature has been around for almost 100 years. But the problem in plant breeding is you've got this inverted triangle where you don't get complete information until right at the end, and then there's no point. So if you can get complete information, as in like a prediction for all of the traits right at the start, your effective population size just goes through the roof. Yeah. I hope that's covered it. There's a lot you could talk about there.
One online. Yep.
So we have a question from Vilu from Simit. So is AGT planning to work on hybrid wheat?
No plans. AGT did work on hybrid wheat varieties before my time, around 2008 or so. So you look internationally at hybrid crops and where they work, like the things that influence whether hybrid crops work are how easy it is to produce a seed. So corn, like canola, you need more systems, but they work really well, right? Seed multiplication rate is massive, especially in canola. So the amount of work that's required to produce the hybrid seed is less than that. And then there's an amount of hybrid vigor. So I think there's definitely like less hybrid vigor or heterosis in wheat, just because because the genome's quite diverse. So there's less penalty there. But even with that aside, like just economically, you've got to produce put so much effort into producing that seed. And in Australia, there's not a very high yield base. So if you're getting like 10% heterosis on 10 tonnes per hectare, that's quite a lot compared to 10% on 2 tonnes per hectare. But probably the biggest reason for us is we've got the endpoint royalty system in Australia, and that means we can capture revenue on breeding whatever way we see best. And we see the best way to breed wheat is through conventional breeding. There's real arguments for it in Europe and other parts of the world where they don't have endpoint royalties and it's harder to capture value. You can mount an argument if you look at the system as a whole to say, like, look, we know if you could fund 2 breeding programs the same and like one program's hybrid and one's conventional, if you could fund them the same, you're better off to go conventional. But we can't fund them the same because we don't have money coming in. So there's people in Europe that are saying, well, we need to create the money through hybrid, run a hybrid breeding program, and overall we'll be able to make more genetic gain because we can fund it better. But that argument just doesn't apply in Australia, I think. So we're very lucky with that system.
I think we might have run out of time for any more questions. Or do we have time for one more, do you reckon, Tim? What's the time? Yep. All right, then. Could you all please join me in giving Adam another round of applause for his time coming out today and explaining a bit of the breeding process. We've also organised just some light nibbles outside of the tute room here. So if you've got time, please stick around, help yourself. Yeah. Thanks again, guys, for coming out for our second seminar.
Thanks.